Hello and welcome. Today we are going to talk about blood. So let's just jump right into it. Blood is super important. It does a lot of things for us. It transports oxygen and carbon dioxide, but it also transports nutrients, wastes, cells, and hormones around the body. So blood is composed of two major components. There is what we call plasma, and then there are what we call the formed elements. So plasma makes up a little bit more than half of the blood. It's mostly water. Also carries a few ions, proteins, and some of our waste products. And then the formed elements consist of erythrocytes, or what we call red blood cells. And their main function is to carry oxygen and carbon dioxide. So they carry oxygen from the lungs to the tissues, and they carry carbon dioxide from the tissues to the lungs. And then leukocytes, which are also what we call white blood cells, and they mostly function in our immune system. The last formed element in blood are what we call platelets. Platelets are small, and we will see some of those in the blood slides that we view, but their main function is in clotting. So red blood cells, or what we call erythrocytes, they are composed of a protein called hemoglobin. Hemoglobin functions to bind oxygen and carbon dioxide, and it binds that oxygen in the lungs and transports it to the tissues, and then binds carbon dioxide in the tissues and transports it to the lungs. So its main function is gas transport in our blood. There are a few pathologies that are worth mentioning. One is what we call methemoglobin. This is what happens when the hemoglobin is in the wrong form, and it compromises its ability to bind oxygen. Also something known as carboxyhemoglobin. This is what happens when hemoglobin binds carbon monoxide, which it has a very high affinity for, more so than carbon dioxide. So if you're ever exposed to large concentrations of carbon monoxide, then it displaces the oxygen that your hemoglobin would otherwise be binding. And so this is why it's important to have a carbon monoxide tester in your house to prevent carbon monoxide levels from getting too high, or at least allowing you to detect them. And then the third pathology for red blood cells is something we call anemia, and there's two types of anemia worth discussing. There's simply having too few red blood cells, which can be determined from what's called a hematocrit, where we determine the percentage of formed elements in your blood. And so if it's below 45%, then that would be considered anemic. Or if there's too little hemoglobin in the red blood cells. And both of these decrease your body's ability to transport oxygen in your blood. Next thing we'll talk about are the white blood cells, and there are two types of white blood cells. There are what we call granulocytes and agranulocytes. Granulocytes have this grainy appearance in the cytoplasm, and they also have lobed nuclei, so that's how we can generally tell them apart from agranulocytes, which we'll talk about in a second. But there are three types. There are the neutrophils, which we see pictured here. Those are very common. They make up about 50 to 80% of our white blood cells. So they have a lobed nucleus with unstained cytoplasmic granules. They are phagocytic, so they engulf and digest invaders or debris in our blood. And we see the levels of these increase if we have an infection. So this is what makes up a large portion of what we would call pus. Then there are the eosinophils. We see one over here. They make up about 1 to 4% of our white blood cells, so a pretty small number of our white blood cells are eosinophils. They have a lobed nucleus with pink stained granules. They are phagocytic, so that's how they function. They also secrete cytotoxic granules that they use mostly when they attack parasites. So these are something that we see increase in number when we have a parasitic response. And then lastly, we have something called basophils, which we see here, this nice little grainy leukocyte is a basophil. It has a lobed nucleus, which can be hard to see, but it has these blue-stained granules. They also secrete cytokines, histamines, heparin, and which is an anticoagulant in our blood. These are notorious for causing inflammation, vasodilation, and they help to recruit other white blood cells. And typically we see these become more active during like an allergic response. And then we have two types of agranulocytes. So agranulocytes lack those granules, and the two types are monocytes, which make up about 2 to 8% of our white blood cells. They're usually recognizable by the large kidney-shaped nucleus and the bluish cytoplasm. This is the most phagocytic immune cell once they become a macrophage. And mostly what they're doing is a lot of cleaning up of dead tissue and bacteria and things. Then the last agranulocyte are what we call lymphocytes. Lymphocytes make up about 20 to 40% of our white blood cells. They're rather small. In fact, they're closer to the size of a red blood cell. You'll notice a bunch of red blood cells around this lymphocyte. They have a large round nucleus with little cytoplasm in the cell. And these function to produce what are called B and T cells, which are a big part of our immunity and our cell-mediated immunity and immune response. Now, it's important to know the different types of white blood cells and what they can be indicative of, because we're going to be doing some white blood cell counts. First, we'll be doing what's called a total white blood cell count, and this basically just tells you how many white blood cells there are, but doesn't indicate type. And as we just looked at, type can tell us what might actually be wrong, as it might indicate whether it's a parasite or an infection or something. And that's what you can gain from a differential white blood cell count. So you'd count the total number of cells and then see what percentage of them is each of the 5 types. And then there's also a net count, which would multiply the differential by the total to give you an estimated number of each. So we'll be doing an exercise with that. I'll point it out in the end of this lecture. So let's talk a little bit now about red blood types. Blood types. These are based on what are called antigens, which is a glycoprotein found in the plasma membrane of red blood cells. There's 3 types of antigens that we're concerned with. Those are A, B, and Rh. And then in our blood, we also have something called antibodies, and so those are found in the plasma of our blood. And antibodies target antigens. So it's important to know what antibodies and what antigens are found in each blood type and how they function if they're mixed. So one thing that's important to know is our blood only contains antibodies for antigens that are not in our own blood or on our own red blood cells. So, for example, if you have type A blood, then you have A antigens on your red blood cells, as indicated by these little glycoproteins. But in your plasma, you produce anti-B antibodies. And so if you have A blood and your blood is exposed to type B blood, then these anti-B antibodies would detect the B antigens and cause it to agglutinate. So if we then look at type B blood has B antigens on the red blood cells, but it contains anti-A. So again, if it was exposed to A antigens, the A antibodies would attack and cause the blood to clump. Type AB contains both antigens on the red blood cells, so it does not contain antibodies for either one. Otherwise, it would attack itself, which is not good. And then type O contains no antigens, but has both of the antibodies. There's one other thing that we're concerned with, and that's the Rh factor. And so you can either be Rh positive or Rh negative. And individuals who are Rh positive have the Rh antigens, and no antibodies against Rh, and individuals who are Rh negative have no antigens, but they produce the antibodies against Rh. So the positive and negative that we see at the end of blood type is indicated by this Rh antigen presence or absence. Now, we develop antibodies against Rh antigens after exposure. So this can be an issue when a mother is Rh negative and a baby is Rh positive. So what happens during the first pregnancy is there are no issues, but during birth, the blood can be mixed, exposing the mother's blood and causing her to produce antibodies against the Rh antigen. And so then if there's another pregnancy, then the mother's Rh antibodies will go into the baby's immune system, causing them to attack the baby's blood. And so to prevent this, there's something called RhoGAM, which is a type of passive immunity that prevents these problems from happening. So we're going to do a little blood typing in lab, and basically what we're doing is we have a patient's type of blood, and we will put that blood into 3 different types of antibodies. And remember, the blood has the antigens, so the antibodies will either attack or not attack depending on which specific antigens they're targeted for. So we put those in, and then we see what happens, and if there's no reaction, then that means those 2 blood types can mix, but also if there's no reaction, then that means that there's no antigen for the anti-A, meaning this blood type is not A blood type. It is B positive blood type, and so we see anti-B antibodies attacking the B antigens, and we see the anti-Rh antibodies attacking the Rh antigen. So in lab this week, we will be doing a computer simulation of blood typing where we will be adding patient's blood to the anti-A, anti-B, and anti-Rh antibodies, and if the antigens are present for those, then that will indicate the patient's blood type. Also, what we'll be doing is we will be counting white blood cells, looking at images of microscope slides, and doing calculations based on the numbers we get. So one thing to know for the blood typing game is follow the link to the website, but you do have to enable Flash. So this is how you do that for Chrome. You would click this little lock button, and where it says Flash, you would click Allow. Or if prompted, make sure you allow Flash. Also, make sure you select Quick Game Random Patients, and then record your results for the 3 patients. For Lab 12, we'll be looking at microscope images of blood. Notice what we're looking at here is just a sample of the total slide. So what's visible here in the large screen is what's in the small red square rectangle over here. And you can increase your magnification using the sliding bar on the left. But notice we'll be counting different types of white blood cells. So if I was looking at this, I'd say this one looks like a neutrophil, and this one over here looks like a lymphocyte. So make sure you reference the previous slides for that. And we'll be putting those counts into boxes and then making some calculations where we count the number of each type that we found, and then the percentage of that divided by 50 times 100, because we'll be counting 50 individual white blood cells. Oops. And then we'll be determining the net count percentage times the total white blood cell count. And so we're assuming there's 10,000 total white blood cells, so we'll multiply those together to determine these numbers, and then make sure you answer your questions in your worksheets, and then also answer the questions in your data sheets. Good luck this week.